I've been at the Broad for 4 years, creating resources, working with diverse ancestry data sets, and I initially started my time here working on the narrow gap data set. Today, I'm excited to be talking about the results of a project I've been working on for the past 2 years. As many of you may know, underrepresented populations can often be excluded from genomic studies, due in part to a lack of resources supporting their analyses. What I'll be talking about today is a resource comprised of the 1,000 Genomes and the Human Genome Diversity Project. And our hope with releasing this data set is that it will serve as a resource to support research of diverse ancestry populations. So before getting into the presentation, I'd like to say that our manuscript is up on bioRxiv, and all of the data set is currently available. In addition to the data I will be showing today, it's available for download here on the Nomad website, and the tutorials that I'll be giving more information on are also available on our GitHub. And also, I'd like to give a big thanks to Mary Johannes, who is my co-first author on this manuscript. In addition to her hard work on the manuscript overall, she also created many of the slides that you will see in the tutorial section. So here's a general overview of the topics that I'll be touching on today. So first, we'll start with a little background on the resource itself, walk through some quality control measures and statistics that we generated, then touch on ancestry analyses, and after take a brief look into some structural variant information, as well as some comparisons of our data set. And then finally, I will walk through the tutorials that we are releasing in addition to the data set, which are for supporting the use of our data set. Starting with some background. Many of you are likely familiar with the 1,000 Genomes Project. Both the 1,000 Genomes Project and the Human Genome Diversity Project are among the most valuable genomic resources because of the breadth of global diversity that they capture, as well as their open sharing policies that allow unrestricted individual level data. Both of these data sets were recently sequenced to high coverage, but they were not well harmonized in the past. And so for this resource, we have harmonized over 4,000 high quality whole genomes from both 1,000 Genomes and HGDP. We've jointly called variants consisting of SNPs, indels, and structural variants, or SVs. We've run variant and sample QC, constructed a resource of haplotypes for use as a phasing and imputation resource, performed ancestry analyses, provided some tutorials for conducting many of the most common QC steps and analyses with these data as well. And all of those topics I will touch on more later in the presentation. So now that we've seen the sample and variant numbers for this data set, here's a general summary of the two data sets and how they interact. So 1,000 Genomes is a good catalog for human variation across the globe, but it is limited in representation. In comparison, HGDP has a broader scope of diversity, but has a smaller sample size, although it does fill in some of the gaps in 1,000 Genomes. For example, it added in samples from the Middle East and Oceania, which were not in the 1,000 Genomes data set. It also fills in huge swaths of samples across African populations as well. Figure B here is from the 2020 HGDP paper on the release of the newly sequenced samples, and it is showing that more variants per individual are discovered within HGDP than 1,000 Genomes. This is likely due to the higher coverage and greater diversity in this data set. So here we can see a map showing where the samples were collected for our across the two projects, as well as a PCA plot, principal components analysis plot. And here is the number of samples. And in the PCA plot, as well as the map, the circles correspond to the 1,000 Genomes project, and the triangles correspond to HGDP, and they're colored by geographical region here. As we'd expect with a PCA plot of global diversity, we see in PC1 that the African populations, colored in purple here, are separated out as the most genetically distinct from the rest of the populations. And then in PC2, we see that the European populations are separated from the East Asian populations, with the Americas, the Middle Eastern populations, as well as Central South Asia and Oceania kind of spread in between those two. And if you're unfamiliar with global PCA plots, this triangular shape is typical to what we'd expect when looking at a global data set. And you'll continue to see this shape as I show more PCA plots later on in the presentation. As I mentioned, our data is currently available for download, and this call set has also already been used as a resource of global diversity by projects Nomad, the Pan-UKB Biobank, the Global Biobank Meta-Analysis Initiative, or GBMI, as well as the COVID-19 Host Genetics Initiative. A primary use of this data is as a global reference for principal components analysis, as you just saw. Our SNV loadings, or single nucleotide variant loadings, are freely shared so that user cohorts can be aligned to the same PC space as our reference panel. And for an example of what that means, here's an example of how GBMI used our data set to align the PC data to jointly look at ancestry when individual level data could not be shared. And so harmonizing ancestry analysis with this resource served as a quality control measure to ensure that ancestral groupings were being applied consistently and that control for population stratification was being performed adequately. All right, so next we will be moving on to the quality control section. So here are the final sample counts for the dataset after quality control, or QC. The quality control of samples was conducted in conjunction with Nomad. Some of the filtering criteria were altered due to the removal of diverse or unique genomes by Nomad sample QC filters. And then the outliers were removed based on subcontinental principal components analysis. We removed PCA outliers as part of quality control, but I will go into more detail on the results of PCA in the ancestry section. So I'd mentioned some Nomad filters were dropping populations. This is an example of that. This type of filter was called NSNP residual, and it was a category of filters. These filters regressed out principal components from the number of SNPs in an effort to identify technical outliers, but they would have excluded whole continental groups, the list that you can see here, and populations as well in this resource, because the groups were distinct from the majority of individuals in Nomad. And this investigation was done by Mary, which eventually led to us adding back in these populations, and Nomad created a minor release with updates to fix the sample filters. So this here is a table which highlights how we dealt with the different populations when joining these two data sets. Ensuring that these two data sets had shared labels across geographic regions made it much easier to run downstream analyses. And this here is a figure showing the mean number of SNVs versus structural variants, or SVs, per individual within each population. And so SNVs are shown here on the top and SVs are shown on the bottom. For the SNVs at the top, African populations had an average of 6.1 million variants per individual versus an average of 5.3 million for out of Africa populations. For SVs, we detected a median of 8,123 SVs in each genome, consisting primarily of deletions, duplications, and insertions. This here is a plot of coverage, and similar to the previous plot, both of these were ones that we conducted after QC that allowed us to get some information on the data set. And so coverage in both data sets is uniformly above 30x, with an average of about 33x coverage across the harmonized data set. And as you can see, the coverage for the HGDP data set genomes is more variable than in 1000 genomes. This is expected based on a variety of technical differences between the two data sets, such as multiple sequencing batches, PCR plus versus PCR free, as well as older cell lines in the HGDP data set compared to 1000 genomes.
Zan, there's a question in the Q&A, and I will ask it now. The question is, were there any math filters set for SVs or SNVs?
So all of the SV calling was done by members of Harrison Brand's lab. I imagine that there were filters, but I do not know what those specific filters were off the top of my head. I would be happy to contact those later if you would like more information.
That's great. Thank you.
Okay, so moving on to the ancestry analyses section. There are a couple different methods of ancestry analyses that we conducted on this data set. This is a list of the 4 ones that we used, though again, PCA was used in addition for QC, so we removed the PCA outliers. These are all methods of visualizing ancestry. Before running them, Mary conducted variant filtering consisting of allele frequency and call rate filtering, as well as LD pruning and relatedness filtering to separate the related individuals from the unrelated individuals. And so this here is showing a larger image of the PCA plot that I had showed previously. When we're conducting PCA, we're not including related individuals, but we do project them into the PC space after the fact. And again, you are seeing that typical triangle that we would expect when looking at a globally representative data set in PC1 and PC2, with PC1 separating out those African populations as the most genetically distinct. That's due to their higher levels of genetic diversity. And then PC2 separating out the Europeans and East Asians, with the other populations kind of split between those two. In addition to global PCA, we also conducted the subcontinental PCA, and this is after quality control. This clustering shows the populations clustering by geographical location, which is better highlighted by the map images that you can see here on the left. And you may notice that in between PC1 and PC2, the Americas populations have less distinct clustering. This is due to high levels of admixture within those populations. And in the quality control process, subcontinental PCA helped us with contextualizing data within continents, as well as detecting the outliers. And this overall signified the need for understanding finer scale subcontinental substructures when conducting such analyses. So moving on to this metric here called admixture. So this figure here, admixture is a program that can be run on data sets. And each row here is showing the breakdown. K is equal to the number of genetic ancestry components that are fit in that run. So that is what each of these are, showing different values of K. We ran through K is equal to 2 through K is equal to 10. And then for columns, although they're not distinctly defined, each column depicts a sample. So for each value of K, there are over 3,000 columns, one for each individual. The lines are very thin. But this shows the individual's regional substructure breakdown. And so, for example, here, when K is equal to 2, the African populations separate out from the rest of the populations, again, due to those high levels of genetic diversity. When K is equal to 3, then we see the European population separating out, and so on. The populations continue to arise as you add in more values of K. And this,
there's another question. I'm sorry, it refers to PCA, so I think I will mention it now, if that's okay. Yeah. So the question is, PCA gets impacted by sample size. So perhaps as sample size increases, OCE may also differ and not be in the same cluster. I'm just curious about the sample size effect.
Can I get some, so if I'm understanding correctly, is that for the global PCA plot then?
That's how I interpret it, yes. And if the speaker, I'm sorry, if the audience member who posted this question is there and would like to unmute to elaborate, please do go ahead, of course. So.
Yeah, if not, would you mind rereading the question again?
Yes, I will do that. So PCA gets impacted by sample size. So perhaps as sample size increases, the OC may also differ and not be in the same cluster.
Yeah, so I mean, when you are, I'm not sure what the exact question is, but the point to which maybe they're referring to the specific location of where the Oceanian populations would be lining up within the PC space.
I think that would be my interpretation, at least.
Yeah.
Yes.
And so I imagine, yeah, if you were to increase, it is the smallest sample size. There are only 2 populations for Oceania overall. It may not cluster in the exact space. We worked with the sample size that we had, so I can't exactly speak to where it would necessarily fall if we were to increase the number of samples.
Of course, that sounds great. Thank you. And again, if the person who raised the question wants to post something further, I'm happy to continue on with that. Thanks, Anne. Please go ahead.
Of course. So then after admixture, 2 other metrics that we used to look at ancestry were F2 and FST. And before I show the 2 plots that result from these, here's a little bit of background on those 2 metrics. So F2 is a metric that results from computing the number of SNVs that appear twice, also called doubletons, and then comparing how often those doubletons are shared among individuals. Since doubletons are rare, computing the number of SNVs that since doubletons are rare, they tend to have risen recently, and thus the results of the F2 analyses gives us information on recent population history. FST is a metric which uses SNVs across different frequencies to calculate the extent of variation within versus between populations. And common variants are used in FST analyses. Those arose a long time ago and thus gives us information on older population history. So this here is a heat map resulting from F2 analyses. Again, detecting recent history based on doubletons. This is also calculated on an individual level, which leads to a much finer scale clustering than you'll see in the FST plot on the next slide. So the brightest part of this map here, these light colors that you can see, is equal to the most doubleton sharing. And this is mostly along the diagonal. And this is comparing, again, the doubleton counts between pairs of individuals. So the column colors here at the leaves of this very clustered dendrogram, these are geographical regions such as, African populations, Oceania, European, Middle Eastern. And the rows here show shades, which correspond to regions for specific populations. And there are 78 populations in total across this entire data set. And as you can see, most doubletons are shared among individuals within the same population and geographic region versus across populations and genetic regions. So you're seeing an African population here that are clustering, an African geographical region here that is clustering within an African population. And you're not seeing much mix in between across those. So here is the heat map for FST. And again, this is detecting genetic divergence from common variants to understand past deep history. FST was calculated on a population level, and you can see the list of our 78 populations here. The higher the differentiation between populations, the greater the FST value. And this results again in a diagonal structure, where an FST value of 0 means there's no differentiation and 1 is equal to the greatest differentiation. You can see how populations end up clustering by region. With the bottom right corner here depicting the African populations, and then surrounding it, we see higher FST values, once again signifying the differentiation between African populations and populations from other global regions. We also do see some populations clustering with inside others, and later on I'll have a larger version of this dendrogram, which you can see more finely how they were clustering. So now moving on to the bit about structural variants and some data set comparisons we conducted. So this here is a graph showing the size distribution of structural variants. Previous structural variant calls in the 1000 genomes data set were already generated before we created our resource. So this figure here is showing only the SV calling done on the HGDP data set. All of the SV calling for this project, as I mentioned previously, was done by members of Harrison Brand's lab. In this plot, we are seeing the characteristic decay that we would expect with more smaller structural variants and fewer larger SVs across the broad SV categories. There are some clear characteristic expectations to this corresponding to mobile elements, insertions, and are labeled here. So in addition to SV calling, there are some dataset comparisons that we did. This was compiling metrics that compared our dataset with each individual dataset that comprised it, as well as the previous phase 3 1000 genomes dataset, which was sequenced to lower coverage, and the widely used NOMAD dataset. I have a plot showing the comparisons, but before we walked through that, I wanted to show each of the datasets that we will be comparing and their coverage, as well as their sample numbers. And so you can see for these 4, they all have above 30x coverage. Our data set has over 4,000 samples with over 159 million variants. Bergstrom HGDP, which is referred to based on the publication, this is the whole genome sequencing version of HGDP, has 929 samples and over 75 million variants. The NOMAD data set has over 76,000 samples, as well as over 644 million variants. This data set here, which is New York Genome Center 1000 genomes, referred to because of where it was sequenced. This is the high coverage whole genome sequence version of 1000 genomes, has over 3,000 samples and over 119 million variants. And then finally, the phase 3 version of the 1000 genomes dataset has 4 to 8x coverage, over 2,000 samples, and over 73 million variants. And so for all of those data sets, this is a plot here that resulted comparing each of the 4 data sets with our HGDP plus 1000 genomes data set. And so you'll note here on the x-axis, this is listed as the minor allele frequency in the HGDP 1000 genomes dataset. And so when you see 0% here, this is noting in comparison data set only. So those and comparison data set in each case is referring to the data set listed at the top, comparing it to HGDP 1000 genomes. So the 0% minor allele frequency means that it does not exist within our HGDP 1000 genomes data set. When creating this comparison, because Nomad now contains both HGDP and 1000 genomes, we built a synthetic subset of Nomad, which removed the alleles counts that were contributed by HGDP and 1000 genomes. And when comparing our HGDP 1000 genomes data set to this synthetic version of Nomad, we showed that variants unique to Nomad were disproportionately rare. In contrast, when comparing the comprising data sets of HGDP only or Bergstrom HGDP, as well as the New York Genome Center 1000 genomes only and the phase 3, our data set uniquely contributes a sizable fraction and number of variants spanning the full allele frequency spectrum, including both rare and common variants. However, rare variants are particularly enriched in all of the comparison data sets aside from Nomad. The HGDP dataset in these comparisons here contains the largest proportion of rare variants. I'll also note that few variants in the phase 3 1000 genomes dataset were not in the HGDP dataset or the New York Genome Center 1000 genomes dataset because the samples were entirely overlapping between those two.
Zan, there's another question in the Q&A. I will read it, and again, I invite the person who's raised the question to jump in if I've missed something. But the question reads: even with 4 to 8-fold coverage, the phase 3 still shows comparative results with the others. And is this surprising?
With the variants, yeah. So that was partially because the samples entirely overlap. We kind of expected to not see much difference between those data sets. I'm not sure if that, I didn't speak to the coverage in particular, but in terms of this comparison here, we weren't expecting anything.
That seems to me to address the question. Thank you very much. Yes.
Okay, so another comparison that was conducted by Lindo, member of the Martin Lab, is a imputation comparison. So this was a comparison for imputation using the phased haplotypes that we released for our data set. We compared imputation quality using different data generation strategies. And this was using a narrow gap data set. So we compared different GWAS arrays versus low coverage sequencing using our HGDP 1000 genomes data set and using the New York Genome Center 1000 genomes reference panel, which is one of the most commonly used reference panels for imputation, as well as TopMed for the GWAS array comparison. This is the largest reference panel available, and again, commonly used. So here is showing the comparison amongst GWAS arrays. We have the GSA array, H3 Africa, and Mega. Our reference panel here is shown by the solid line. And when imputing GWAS arrays, you have lots of options for reference panels because you don't need individual level data. You can see here that our reference panel outperforms 1000 genomes alone, but it does do worse than TopMed, which we expected due to the much larger size of TopMed compared to our data set. Our future plans do include phasing our 1000 genomes and HGDP data set with the Nomad data set. And then this figure here is showing the comparison of low coverage whole genome sequencing. So for low coverage, one needs individual level data. So we were not able to use TopMed in this comparison. And across all coverages, our reference panel performed better than 1000 genomes. And for variants under allele frequency of 1%, including HGDP in the reference panel was equivalent to doubling your sequencing depth. All right, and so moving on to the last tutorial section here. So this is one of the parts that I am a bit excited about as someone who likes creating resources and such. So in addition to releasing this data set, to increase the accessibility and facilitate future projects, we've put together publicly available tutorials that use cloud computing to conduct common genetic data analyses, and these are implemented primarily in Hail. They're located in our GitHub that is linked here, and I'll continue to link throughout the slides if anyone gets curious and would like to take a look. So here's an overview of the tutorials that we've created. These are 5 IPython notebooks, including QC of sequencing data, relatedness inference in PCA, calculating statistics by population, analyzing genetic divergence, applying ancestry analysis to a new data set, using the HTTP 1000 genomes as a reference panel. And so when creating these tutorials, which were primarily created by Mary and I, we wanted to have a set structure across the notebook so that no matter which notebook someone looks at, they're able to kind of get their bearings easily and they're easily navigatable. So we have a titles and authors section at the top. There's a setup warning in case anything that you need downloaded on your, when you start up your cloud cluster, you might get an error. So we'd like to warn users that they need those. There's an index using clickable links to make it easier to move through the page and get to sections that you may be interested in, as well as including a general overview, which gives the overall purpose of a notebook and description of analyses run. So if you're not sure what a specific notebook does, just based on the title, you can read through the overview and get an idea and see if a step that you'd like to learn how to do is within such notebook. We also, of course, include all the libraries that we import and try to set up the default paths in a way that's easier for users to go in and change to what data sets and output paths they would like. So here I'm just giving an example of what a title, author, setup warning, and index section would look like. The specifics aren't necessarily important, but all of them have this similar look. And then this here is kind of the general overview section. We also link to, relevant papers, explanations of abbreviations that we use, as well as linking to, of course, the projects that we retrieve data from. Here we're showing, importing all of our libraries and then the setup of default paths that we use. So then moving on to walking through the individual notebooks one by one, we'll start with notebook 1. So this notebook focuses on metadata and quality control. So in this notebook, we're applying Nomad's quality control filters. We create plots using different metrics before and after QC. We identify those Nomad sample QC filters, which are removing certain populations, and we retrieve them. And then throughout this process, throughout all those steps, the user also learns some useful Hail methods, namely reading and writing a matrix table, as well as filtering a matrix table, using both an external imported function, as well as filtering with a list of samples to remove. So this here is just to give an example of one part of notebook 1. This figure here is just showing the function that we use to apply Nomad's QC filters. We additionally, earlier on in the general overview section, linked to Nomad's webpage where more information on the specifics of their QC pipeline can be found. And this one here is just showing a plotting function that we used to plot the number of SNVs. And these 2 plots here are the result of that function. And so for multiple metrics, we're looking at the numbers pre and post QC to kind of get a better look at the data set. So moving on to notebook 2. So this notebook focuses on PCA and ancestry analyses. We started those by doing some variant filtering as well as LD pruning. And then we ran Hail PC Relate, which then resulted in separating into 2 data sets of related and unrelated individuals. And as I had mentioned previously, then we ran PCA on unrelated individuals and then projected related individuals, and finally resulting in removing 24 PCA outliers and rerunning PCA after that. So once again, taking a quick look kind of inside of notebook 2, these are 2 components of the overall notebook. This is showing how we ran PCA on unrelated individuals and then projecting related individuals. And these are the resulting PCA plots. You may notice these are a bit different than the PCA plots that you had seen earlier on in the presentation. These are the plots using Hale's methods. We also did include PCA plots using R. And so this is right now, R currently has a bit more robust plotting options. And so this is what we used to generate plots for the manuscript and also the plots that I've been showing kind of through the course of this presentation. So now moving on to notebook 3. In this notebook, the idea was just to kind of do some summarizing of the data post quality control and some of the results of PCA. So we went through and selected the columns that we needed for a post QC summary table. Then we annotated that table with some relatedness information, then went through and calculated certain statistics per population, and finally formatted that into a table and exported it into a TSV, which was used to create some plots, such as the number of SNVs per population that you saw previously, as well as that coverage plot. And so here is an example of what happens. This is specifically calculating statistics per population. You can see in these notebooks, we also include more information on specific Hail functions. So if you're unfamiliar with Hail functions, this is a drop-down menu that will have a clickable link, and you can it will take you to the Hail documentation page where you can learn more about whatever functions that we are using. We also include some outputs from tables so that you can check on your work as you are continuing to run through the notebook. And as I mentioned in this notebook, we do use relatedness information from the previous notebook, but you don't need to run all the notebooks in one line. You can run them individually on their own. And so now moving on to notebook 4. So this notebook focused on F2 and FST and kind of separated between those two metrics. So for F2, first we filtered to unrelated samples and then separated out the doubletons, ran a call rate filter, and then compiled a set of samples with the doubletons, and finally recorded the shared doubletons between sample pairs. For FST, we first again filtered to unrelated samples and then exported to Plink, then created a list of all population pairs. We ran FST using Plink, and then finally recorded the mean FST estimate for each population pair. And then so here's a screenshot of code from notebook 4. At the top here, this is showing how we obtained the samples that had the doubletons, as well as this is a screenshot of the FST calculations and the Plink command that we used to run such FST calculations. In this notebook, there were also additional analyses that we ran for FST, and this was using R. So I had mentioned that we had a more detailed dendrogram from the FST analysis, and so that is shown here. You can see this is zoomed in from the FST heat map that I had showed previously, and this allowed us to find those interspersed populations. You can see that within like this somewhat larger cluster of European populations, there's some Middle Eastern populations clustering. And the main takeaway from this dendrogram overall is that ancestry is a continuum. And then in this figure B here, this scatter plot, this is showing the genetic differentiation measured by FST versus geographical distance. And there is a linear relationship. There are differences that you can see by project. Specifically, the HGDP project has a steeper slope relating distance to FST, and this is likely reflecting the broader anthropological design intended to capture more divergent populations compared to the samples in 1,000 genomes that reflect some of the largest populations. So then finally moving on to notebook 5. So this notebook focused on assigning ancestry labels using a random forest model. So to do that, we intersected two data sets. In this case, it was our HGDP 1,000 genomes as well as the Gambian Genome Variation Project, or GGV, then applied a random forest model, which was trained on population metadata labels from Nomad to learn population labels in the new data set that we intersected. And then we went and built a random forest model with unrelated individuals from the HGDP 1,000 genomes data set, and finally created PCA plots after applying the random forest model to the GGV data set. And so here is just a quick look inside of notebook 5. This is showing kind of one of the functions used to intersect with the reference data set. And this slide here is showing those resulting PCA plots using our HGDP 1,000 genomes as a reference. Here, the GGV data set shown in gray, so you can see where it clusters in comparison to our data set. And so now that we've walked through all of the notebooks, I'd like to kind of show some video of navigating through the notebooks that we have. So this video here is showing the README that we have on the notebook. We have packages installed as well as how to start your Google cluster. And then as you can see, clicking that link will bring you to Notebook Viewer, which is what we suggest using in order to look at our notebooks. It allows the clickable links to be functional. Unfortunately, if you go on the GitHub and try to use our clickable links, they don't work properly within GitHub. You can still look at the notebooks. It's just the navigation isn't quite as easy. We also include the runtime indicated across notebooks as well as validity checks. So if you're running through them yourself, we include what number of samples or variants you might expect after certain steps. So if you're running this on your own, you can make sure that your output is correct. And so here's another thing to highlight. So this is showing in the notebook viewer, we have graphs included, and this is the number of SNVs that I had shown previously. You can select or deselect certain populations or metrics that you would like to see within plots. This here is also showing kind of navigating using the index of the notebooks. And again, showing here how you can hover over specific points and you can see what their values are, what populations they are assigned to in this case, and you can hide or show metrics depending on what you would like to see within the plots. I think this is a useful tool when running analyses to be able to get a better idea of working with your data set. And so in summary, this resource of HTTP and 1000 genomes is a diverse data set with publicly available individual level whole genome sequencing data. It's currently available for download on the Nomad website, and our code as well as the tutorials that we walked through today are all available on GitHub. And with that, I would just like to give a big thank you to all my collaborators, especially Mary and Lindo, as well as Alicia Martin. Thank you.